If an organism's genome is going to remain constant in size, it means that gains in genes have to be balanced by losses in genes. Gains in DNA have to be balanced by losses in DNA. These gains and losses can be described as more conservative versus less conservative, or more radical versus less radical. Less radical changes include point mutations. This introduces new information into genomes, although it doesn't necessarily change the genome size. Orthologous replacement, where you're swapping one gene for another gene. It's new DNA or genes coming in, but it's not also changing the size of the organism. Or gene duplications, which in fact do change the size of the organism, but don't actually introduce new information into the organism. In terms of more radical or less conservative changes that introduce new things into genomes, illegitimate recombination, where you're having insertion of new DNA types, the insertion of prophages into genomes, the acquisition of plasmids, the acquisition of genomic islands, and also the acquisition of endosymbionts are ways of increasing the DNA content of cells by substantial amounts. On the outside, point mutations are balanced by point mutations. Point mutations not only introduce new information, they take away old information. In orthologous replacement, when you're swapping one gene for another, you're gaining a gene, but you're also losing a gene, or gaining DNA and also losing DNA. In terms of more radical changes, you have deletions that remove information from chromosomes, also reducing chromosomes in size. In fact, this is the primary means by which chromosomes are reduced in size. You have pseudogene formation, which is basically taking genes that otherwise would be functional and converting them into genes that are not functional through mutation. And then you have overall genomic erosion, which is essentially a combination of these 2 points, where the genome becomes reduced in its functionality as a consequence of reductions in its genetic repertoire.